Okay, well, good afternoon and welcome to our webinar, Smart Medicines, the Future of Targeted Therapeutics. My name is Professor Ruth Cameron. I'm one of the directors of the Cambridge Centre for Medical Materials at the Department of Materials Science and Metallurgy, where our multidisciplinary research concerns materials that interact therapeutically with the body. This session is part of the Future of Physical Sciences webinar series, which was launched to keep alumni and supporters connected to the cutting-edge research taking place here in Cambridge. Research that's deepening our understanding of critical global challenges and helping us to shape how we respond. Today, we're going to be focusing on how advances in material science and chemistry are revolutionizing medicine, creating treatments that can sense, respond, and adapt inside the body to improve patient outcomes. We're joined by three outstanding speakers, Professor Serena from the Department of Materials Science and Metallurgy, Professor Melinda Duer from the Yusuf Hamied Department of Chemistry, and Professor Gonçalo, also from the Yusuf Hamied Department of Chemistry. Each of them is going to share how their research is driving innovation and shaping the future of targeted therapeutics, opening up new possibilities for smarter, more effective treatments. So I've just got a few things to say around housekeeping. After the talks, there's going to be time for your questions. So please submit them at any time using the question and answer button at the bottom of your screen. We're going to address as many of those as possible, and we'll do that after all the talks have finished. So we're going to move on to our first speaker. This is an exciting time for medical science and for all of us who are thinking about the future of healthcare, the future of patient outcomes, and the technologies that are going to transform treatment. So let's just dive into the science, the challenges, and the work underway here at Cambridge to better understand a rapidly changing world. Our first speaker co-directs with me the Cambridge Centre for Medical Materials, and it's my pleasure to introduce her. Over to Professor Serena Best.
Thank you very much. As Ruth said, then we run the Cambridge Centre for Medical Materials together. And although we cover quite a lot of different materials, today I'm just going to focus down on the things that we're doing with collagen and our development of collagen structures for tissue models and also for regenerative medicine. So just a bit of background about our groups. We have a multidisciplinary group, and so we're really interested in combining the skills of everything from surgeons to electrical engineers and materials scientists to help us to develop our collagen scaffolds and implants. We have a range of different applications, and these have included cardiac tissue repair, nerve regeneration, dermal repair and wound dressings, thinking about orthopedic applications, dental applications. And we're also interested in not just the implants, but thinking about developing substrates for bioreactors. So, for example, we were interested in looking at platelet production, but also developing model structures for drug testing. And so here we've been working in the fields of mammary tissue, and more recently we've been moved into organoid structures, so applications including the lung, the kidney, and the endometrium. So when I'm talking about collagen scaffolds, I should perhaps take a step back and explain how we're actually making these structures. So basically we have our end product, which is almost like a little sponge, like a bath or a kitchen sponge. And the way we make these is to take insoluble collagen 1, and we will suspend that material in say a beaker. Forms ice crystals, and the ice crystals are pure water, and so they reject the collagen to the positions around the ice crystals that are formed. If we then reduce the pressure and then we increase the temperature again, then we can induce ice sublimation to happen. And so that's where the ice moves straight from the solid state into the gaseous state. And so all of that gas escapes, and we are left with the negative of our ice crystals, and we have our porous scaffold. What we can then do is think about things we can do to control various aspects of these structures that we've produced. And obviously, we're interested in creating interesting architectures. We can control the biochemistry, and we can also look at the physical response of these things that we've made. And all of these are going to influence the eventual biological response. So, as I mentioned, we're using lyophilization or freeze drying. And so, when we're cooling our material down, then we're going to be using a set cooling rate, and then we're hoping to achieve a final set freezing temperature. So we can achieve this using sort of conventional commercial freeze dryer. But what we also need to think about is not just the set temperature that we're asking the freeze dryer to achieve, but actually what's going on inside our mold or our specimen. And so what we do is we put thermocouples into the mold, and as we cool the suspension of our collagen and our water, then eventually we'll reach the freezing temperature. But what happens as the crystals are being produced is we have a latent heat associated with that freezing, so as heat released. And so the temperature increases. There's then a holding time, a freezing time before the temperature begins to decrease again. And using this process and our knowledge about the nucleation and growth of ice crystals, then we found that we can control the pore size within our structure to quite a considerable degree. So we can produce porous structures with anything from pore diameters of around 80 microns right up to 200 microns. And this is important because depending on which particular biological cells we're interacting with, then we may wish to have different scales of structure. The other thing we need to think about is not just the size of the pore, but we need to consider the size of the interconnections between the pores, because if we want cells to be able to migrate through the structure, then it's important that they need to get from pore to pore. And so what we found is that we can put cells on the top of our scaffolds. And even if you have a very large pore size, if you don't have sufficiently large interconnections, then the cells are going to get stuck. So they can't migrate and fully fill up the scaffold. And we can define a percolation diameter. So we can think about the distance that a notional hard sphere can travel through our structure. And depending on how far that solid sphere can travel, then this will allow us to create a graph. So we can think about the relationship between the distance traveled and the structure, and we can define a percolation diameter. And clearly, we want to have sufficient interconnections, or they have to be sufficiently large that cells can move throughout the structure. But the other thing we can do is think about the isotropy of the structures that we have. And so once again, I'm thinking about the way in which we're cooling and processing using freeze drying, and thinking again about the use of these thermocouples. And what we find is that depending on how much we're filling up our molds with our suspension, then we can create areas of the scaffold that have completely contrasting architectures. So when we're thinking about the structure which is occurring near to our mold base, then we find that it's fairly isotropic. But as we move away from the mold base, then we find that the there's a dendritic growth of the ice crystals, and so that means that we end up with very aligned collagen pores, structures within our scaffold. So you may wonder why that's of interest. Well, if we begin to think about the tissues that we have in our body, then very few of them are truly isotropic. And so what we find is that the growth of our very aligned structures is actually quite useful for cardiac tissue repair. And with our colleagues, so Dr. Sanjay Sina, then we were able to look at introducing cardiomyocytes into our scaffolds and trying to create structures that were truly reflecting the cardiac tissue. So far, I've just been talking about architecture, but clearly we need to think about the biochemistry and the physical response. But in many respects, these are interconnected. And this is due to the processing that we might want to make after making those structures. And that is thinking about chemical cross-linking. So we're cross-linking our collagen fibers in order to perhaps give the material better strength or better resistance to applied forces. And so this is great. We can produce stronger and stiffer structures. But actually, what we have found is that when we do this, then the cross-linking that we're inducing is actually using up some of the integrin binding sites that the cells might be looking for. And so if we have a look at how the material is or how the cells are reacting in contact with the substrate, then if we apply too much cross-linking, then we can actually ablate, we can switch off cell binding altogether. So you might think, well, that's probably going to be a bad thing. And in many respects, it might be, but actually we can make use of that. And so what we're able to do is to switch off all of those binding sites if we choose to by using very high degrees of cross-linking. And then with Richard Farndale and his group and the Department of Biochemistry, we found that we were able to attach triple helical peptides onto the surface of the collagen and actually offer very specific functionality to the scaffolds that we were producing. So moving on, other things we're doing with our scaffolds and our collagen surfaces is to think about angiogenesis. So this is the production of blood vessels, which we are hoping are going to move throughout the scaffold. And so here we're interested in using co-cultures, so thinking about using human osteoblast cells and human dermal microvascular endothelial cells. And the combination of those actually leads to the production of blood vessels. And these will supply nutrients and oxygen throughout our scaffold. If we don't have angiogenesis, we may end up with a dead core to our scaffold where cells might have migrated to but are not receiving the nutrients that they need. And then finally, another example of how we're using our scaffolds as tissue models. We've been working with people in for example, Teresa Sindrova Davis and Kathy Nyakin, and we're looking at endometrial organoids and how we can put those into structures that are reproducing as much as possible the environment of the endometrium. And here we found that rather than just using scaffolds on their own, actually by filling our scaffolds with gels, we're producing an environment where the embryos are actually able to attach and sort of work their way into the structure. And so we're looking at endometrial organoid fragments, and we're looking at those in our collagen gel combinations.
Thank you very much, Serena, for that great overview. So just a reminder to everybody that we will be taking questions at the end when everybody has spoken, and you can enter those questions into the question and answer function on the Zoom thing. I'm collecting those as we speak. But for now, let's move over to Melinda Duer. Over to you.
Okay, so good afternoon, everyone. I'm very delighted to have this opportunity to tell you something about the research that my group and I have been doing over the last few years, and particularly in the area of developing new cancer therapies and where I think we're headed on that. Along the way, I'm also going to tell you a little bit about some of the new techniques and methods that we've had to develop in order to answer some of the key questions that you need to be able to know the answers to in order to do this kind of work. So I'd like to start by introducing you to one of your tissues, and Serena's pretty much done this for me already. But if you drill down into a tissue with a microscope, what you see, of course, is that tissue has cells in, as Serena's been describing. And then around those cells is this material, this extracellular matrix. And primary in that extracellular matrix are the collagen fibers that Serena's been talking about, it's protein collagen. And in between that, there's a whole load of glycans and sugars like hyaluronic acid and chondroitin sulfate. As Serena's already told you, this extracellular matrix is the material that essentially is giving your tissues their shape, and it gives your tissues their mechanical properties. So it's what makes bones stiff, it's what makes tissues stretch like skin. But for decades, the extracellular matrix has been viewed as simply a passive material. It is just something that gives tissues their structure. But what if it was more than that? And Serena's already implied that. What if it's more than that? What if, in fact, the structure of the matrix is actually directing the cells in how to behave? Now, we know that the extracellular matrix composition is highly specialist for every tissue. We know, for instance, that bone cells only behave like bone cells when they're in bone extracellular matrix. And we know that the extracellular matrix changes drastically in disease. And it changes particularly in cancer. One of the first things that change in cancer is the extracellular matrix around the tumor. And I'm just going to pull up a laser pointer there so that you can see where I'm pointing to. It's this material, this extracellular matrix around the tumor is one of the first things that change as soon as the tumor cells start to develop. And there's increasing evidence, I think, now that those extracellular matrix changes correlate with disease progression and moreover potentially actually are causal in driving disease progression. So suppose we could understand how the extracellular matrix instructs cancer cells in enough detail that we could restructure the extracellular matrix and tell cells how to behave more like normal cells. And why we took that idea forward, the big driver behind our work, is that medicine needs every possible therapeutic route to treat cancer, to defeat cancer. The migrating cells, the cells that peel away from the primary tumor and investigate through the extracellular matrix and set up metastatic tumors elsewhere, these are not treated by most of the current treatments for cancer. And it's, of course, these migrating cells that are the most dangerous ones because these are the ones that are responsible for cancer spreading. Now, on top of that, the existing toolbox of drugs have a number of profound issues they have to deal with, particularly drug resistance, for instance, and the fact that those drugs are having to treat a huge variety of different cancer cells, even within the same tumor. Because in any given tumor, there are many, many different phenotypes of cancer cell, and typically a drug will have a different effect on each of those cells. Each of those different cancer cells has a different sensitivity to the drug. And so whilst you might kill out some subpopulations of cancer cells, others will remain behind to progress the tumor. And now, of course, they've got more space and nutrients to do that. And then on top of all of that, if this extracellular matrix is constantly telling cancer cells, invade, 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 but medicine only has half a toolbox. It can't stop the matrix from doing that. So it really wasn't a difficult question or a difficult decision for us to make to decide to work on how the extracellular matrix directs cell behavior. The first problem is how to study the extracellular matrix. It's a highly heterogeneous, complex 3D architecture. So the first thing we did was we developed nuclear magnetic resonance or NMR spectroscopy methods to study the molecular structure of extracellular matrix in native tissues and in tissues that we can grow in the lab. And this is Dr. Wing-Ying Chow, who did much of that work. And as you can see, she absolutely loves it. What NMR gives us are spectra like these, and these are essentially fingerprints of the molecular structure in the extracellular matrix of whatever tissue it is we've put in to the spectrometer. So here we're looking at the molecular fingerprint of collagen in mouse bone, and we're comparing it with the molecular structure of a bone-like tissue that we've grown from cells in the lab. Now, being able to compare the molecular structures of the matrices in two different tissues meant that we were able to then rationally refine how we grew our lab-grown tissue to get a tissue that was demonstrably highly similar to the real in vivo tissue. Now, we need lab-grown tissues to be able to follow biological processes like how do cancer cells invade into the matrix, into the tissue. And we need those lab-grown tissues to be highly, highly similar to the real in vivo tissues if we want to be able to translate any of our results from the lab into humans and to develop new drugs. So what I'm telling you about here, which actually took about 10 years, is actually a fundamental key step for everything that came later. So roll on about 20 years, and we have much more insight into how the extracellular matrix structure changes in cancer. How do we now turn that new understanding into new cancer therapies? Well, I would strongly advise that the first thing you do is you have excellent researchers in your group, and I think everyone on this call would attest to that. So Yulia here, Rakesh, and Agna, everything I'm about to tell you is primarily down to them. So the first question we had to answer was, how do cells take their instructions from the extracellular matrix on how to behave? This is absolutely fundamental to the question of how to stop cancer cells from invading, because it is something about the extracellular matrix that's telling cancer cells, effectively, they're clear for takeoff. They're able to move and should move and invade. What was known at this point was that cells interact with the extracellular matrix, and Serena's implied this, shown this already, through extracellular matrix molecules binding to specific proteins, receptors on the surface of cells. And that extracellular matrix receptor binding sets off a whole cascade of chemistry at the other end of the receptor inside the cell. And that chemistry is what defines the particular cell behavior that then happens. But the key thing is that sometimes these matrix receptor interactions seem to happen, and sometimes they don't, even when they're both present in the tissue. And sometimes they happen and cause a huge cell response, and sometimes they happen and seem to do hardly anything. So it's really, really unclear exactly how the cells are detecting the matrix and what it is they're detecting about it. And we absolutely need to know that if we're going to be able to use the matrix to reprogram cells, and particularly cancer cells. So we decided to work on how the extracellular matrix directs cell behavior in brain cancer, specifically glioblastoma. And we decided on glioblastoma because primarily it's a highly aggressive tumor and there are very, very limited treatment options. But it was also because the glioblastoma tumor matrix is absolutely stuffed full of fibrous proteins. And that's a very, very different composition to the extracellular matrix in normal, healthy brain. And so it seemed to me that if the aberrant extracellular matrix is directing, say, cancer cell invasion anywhere, we should see it in glioblastoma and then potentially be able to do something about it. Now, when you're working at the interface with medicine, as we clearly are here, it's absolutely essential to engage with the top clinicians who see the treatment challenges firsthand in order so that you can test and shape your ideas. Dr. Richard Mayer here, he's both a fantastic research scientist here in Cambridge and a neurosurgeon at Addenbrooke's. And it's down to a lot to him that the many, many discussions I've had with him over the last few years have really led us to where we're going now. So we knew from the work of others that one particular molecule in the extracellular matrix of brain had been implicated as being likely a contributor to tumor progression. And that molecule is hyaluronic acid. So we looked at whether hyaluronic acid could trigger glioblastoma cells to proliferate or migrate or do anything really to progress the tumor. And so what we did is we grow these small tumors in the lab from patient cells, and then we immersed them into a gel containing hyaluronic acid. Now, when that hyaluronic acid concentration in those gels is similar to normal brain or higher, we see very, very limited migration or invasion of the tumor cells into that surrounding brain-like matrix. But then what we did was we diluted the gel to mimic the increased fluid that will always be in and around a brain tumor because of the inflammation that happens. And what we saw then was this huge migration of cancer cells into that matrix. Now, to start with, we thought that was simply a viscosity effect. If you dilute the gel, of course, you're making it easier for cancer cells to move through the gel because it's less viscous. But that turned out not to be the case. Rather, what we were seeing seemed to be, in effect, this increased cell migration here seemed to be driven by the presence of hyaluronic acid. But totally bizarrely, it looked like the more hyaluronic acid is present, the more the cancer cells were shut down and told by the matrix not to migrate. So we were really struggling to understand why when you put more hyaluronic acid around a cancer cell, it seems to shut down the cell invasion, and yet hyaluronic acid is essential for cell invasion. And then one morning on my bike, I had one of those Duer moments and realized that the main difference between hyaluronic acid in high and low concentrations is the flexibility of the hyaluronic acid molecules. And to trigger any reaction in a cell, the first thing hyaluronic acid has to do, of course, is bind to its cell surface receptor, like any other extracellular matrix molecule. And to do that, it was clear the hyaluronic acid molecules have to bend and flex in order to be able to reach those receptors down on the cells. The hyaluronic acid molecules bend and flex naturally through thermal motions, but I think you can see they can move much more when there's edema, when there's fluid associated with inflammation around them, because of course that extra fluid pushes the hyaluronic acid molecules further apart, and so they simply have more space to move. So we used NMR spectroscopy to measure the amplitude and speed of the molecular motion of hyaluronic acid molecules in these settings. And what we found was that only when hyaluronic acid molecules can bend through more than 25 degrees on the nanosecond timescale do they ever trigger response and trigger invasion. And that is because the hyaluronic acid molecules have to bend that much in order to dive down to the cell surface and bind effectively onto their cell surface receptor. So the question then is, can we change that signal that the extracellular matrix is giving the cancer cells here? So if it takes a flexible hyaluronic acid molecule to trigger cancer cell invasion, then how about we present cancer cells with some immobile hyaluronic acid molecules? Could that prevent this cell response to invade? So to do that, we took advantage of the fact that these brain tumors are surrounded by these very stiff fibrous proteins, essentially immobile molecules, and we made a hyaluronic acid molecule with some chemically modified groups that we could chemically click on or clip on to that fibrous matrix, thus creating essentially immobile hyaluronic acid molecules just at the cell surface where the tumor proteins are around the tumor cells. And when we did that, it completely shut down the tumor invasion into the surrounding gel. Now, this cross-linkable or clippable hyaluronic acid is not toxic, it's not killing the cells. It reprograms them to behave more like responsible citizens rather than hoodlums. Now, as far as we've been able to tell so far in animals, this drug has very little effect on the normal parts of the brain that don't have this fibrous matrix, or because they are already full of, effectively, immobile hyaluronic acid. By adding this extra chemically modified hyaluronic acid, we're simply presenting normal brain cells with their normal environment, so it doesn't change their behavior at all. So this drug won't remove a brain tumor, but we're hopeful that if it's infused into inoperable tumors, for instance, in the brain, it could seriously slow down tumor progression with hopefully fairly minimal side effect, because as I say, it's non-toxic. I think that it can certainly be used during tumor resections to shut down the cancer cells that are inevitably left behind after surgery, because the surgeon can never remove the whole tumor in the brain because they have to remove too much of the brain. And for me, the beauty of this approach is that we're treating the whole tumor. We're making the tumor environment itself tell cancer cells not to invade. And so I'm now certain that the extracellular matrix is a master regulator of cell function, and that in all diseases, but especially in cancer and degenerative diseases, it's effectively defining how the disease progresses. It is that damaged and altered extracellular matrix which typically precedes the aberrant behavior of cells that we see in all of these neurodegenerative diseases, in aging, and of course in cancer. So what we now need is a paradigm of drugs to treat aberrant extracellular matrix. all the drugs that are currently trying to restore normal behavior for cells in degenerative diseases and in cancer are trying to do that by getting inside the cell and changing the chemistry that's happening there that's driving the poor cell behavior. But if it's the extracellular matrix that's fundamentally causing that poor cell behavior, then we need to stop the thing at source. We need to stop the disease by altering the extracellular matrix. And if we can do that, then we can give medics a full toolbox of drugs to treat cancer. So my group's still got a way to go to putting that extracellular matrix modifying drug into the clinic to treat glioblastoma, but I'm now certain that it can. And as I keep telling everyone, I'm not going to retire until we've done just that. And then finally, this is the rest of the group. Last year, we haven't got around to taking a photograph this year for a whole load of reasons, but I'm delighted to have told you about what we're doing. And if you'd like to know any more, please do get in touch. We're always very pleased to field questions, debates, and of course, at the end of the talk today.
Thank you very much, Melinda. Some amazing food for thought there. Now over to Gonçalo Bernardes, and look forward to hear what you've got to say.
So thank you so much. I'm really delighted to be at this alumni event. And my research group, we are particularly interested in using chemistry principles to develop new medicines whose action is restricted to the tissues or to the cells where they should work in order to maximize therapeutic efficacy and reduce side effects. There are different angles by which we are approaching this quest, one of which is where we are developing drug conjugates, where we conjugate linkers and payloads, so cytotoxic molecules, to new shuttles. Those new shuttles for the specific tissues are de novo computational design mini binders, and this is a topic we are really pushing and is now entering into the clinic. But today I will focus on a different class of molecules, and this is really the development of what we call RNA degraders or RNA scissors. These are small molecules that chyme, natural products, and ribonucleases are capable to irreversibly degrade RNA. This is a new field in my research group, and it was in 2020 that we disclosed the rational design of click degraders. In this slide, I would like to show you the two mechanisms by which nature degrades RNA. One is using these enzymes that are called ribonucleases. They come into close proximity of the target RNA, and there the enzyme is able to deprotonate this 2OH prime in the ribose of RNA. This is a major chemical difference between RNA and DNA. It's present in RNA, but not in DNA, and therefore and thus there is this inherent instability of RNA, because you can deprotonate quite easily this 2OH prime, and this then leads to the cleavage of the phosphodiester backbone and cleaves the RNA. The other way that nature uses to cleave and to degrade RNA is, for example, highlighted by the mechanism of action of this natural product. This natural product has RNA binding motif, so it binds to a particular RNA. It's called bleomycin, this natural product. And then it has this imidazole moiety that can grab transition metals in the cell to locally produce reactive oxygen species. And our approach was very simple and very naive, perhaps because we were not experts in this field. And what we designed were these click degraders, or these RNA scissors that are very simple. At this time, they were a simple imidazole with a linker and a clickable handle, something you could covalently attach to RNA and then ask the question, if I covalently attach an imidazole moiety to RNA, does that lead to degradation of that particular RNA? And the long story, making it really short, is yes. We first applied this to in this way. So we have an oligonucleotide where we can install a clickable handle in one of the bases. We do our click reaction with the imidazole scissors or degrader. And what we have observed is that if we click this imidazole into the RNA becomes unstable and degrades over time. And we have used this information to develop a method that we can probe certain RNA modifications. For example, one modification that is linked to cancer that is called M6A. So it's a methyl modification at the sixth position in adenosine. And this we published well. And while this was important to validate the approach that we can use small molecules to either deprotonate the 2OH prime or either to locally produce RS species and degrade RNA. And this was at the time more as a molecular tool to look into RNA modifications. And then we asked the question, what if we could, instead of covalently link the RNA scissors, so what cuts the RNA, the imidazole in this particular example, what if we could bind it in a non-covalent manner using a small molecule to degrade that particular RNA? And that's what I will show you to you next. Just as an introduction, our genome, so from the old human genome, only 1.5% actually of our genomes produce protein. And from this 1.5% that produces protein, very few proteins have been used as targets for pharmaceuticals and for the development of new medicines. And from the number of proteins that the genome encodes, there is a large number that are the so-called non-drug or undruggable proteins. So one particular interesting view or one interesting approach is that in our human genome, there is this large portion, around 70% of non-coding RNAs, that in principle could be drug targets. There are a number of different approaches that people are taking now. One is the use of, for example, antisense oligonucleotides, where you have a complementary sequence to the target RNA. However, it's very difficult to develop these antisense oligonucleotide drugs. And apart from the ones that target the liver, there are very few examples of successful development of this modality. Then you have small molecules, like what we have been learning with proteins. Typically, a protein has a function. We inhibit that protein, and we block the function. So there is a direct readout and the function that we are preventing that protein from doing. But with small molecules, when they bind to RNA, usually not so much happens. Now, there are very few functional sites on RNA, sites that you can bind and stop, for example, translation. So our idea was pretty simple again. It's the development of these actual bifunctional molecules. We call them PINADs, for proximity-induced nucleic acid degraders. And what we try to achieve is that we would use an RNA binder that binds to a particular RNA of interest. And then once it binds in a non-covalent manner, and the scissors or the warhead would locally induce degradation of that particular RNA, only in the cells where that RNA is important. So this will be an autonomous process. This don't require any enzyme, anything that is in the cell. And this will not require, for example, formation of ternary complexes with enzymes that are then needed to do the degradation process. And we could design these molecules by the principles of medicinal chemistry. So this is the first binder, or one of the binders that we have now advanced mostly into very late preclinical stages. It's a strategy where we are using these DPAs or D2-picolylamine warheads or scissors. How do these work? These ones here focus exclusively on the mechanism of bleomycin, the natural product I showed you before, because it binds to transition metals in the cell and can locally, in a catalytic manner, produce reactive oxygen species. So how does this work? So the binder binds to the target RNA, and then locally the warhead grabs transition metals in the cell, for example, copper, and I will explain why copper is so important in this story, generating in a localized manner reactive oxygen species that would lead to degradation of the RNA. So does this work, and what was the proof of concept that we have been generating? So we have been particularly interested in copper and oxidative stress because in cancer and neurodegenerative diseases, these cells are known to undergo a process that is called euproplasia. And this is the dependency of cancer cells and cells undergoing neurodegeneration on copper for those particular two biological processes. And typically in those cells, in cancer cells and in neurodegenerative conditions, these cells are undergoing oxidative processes in a much faster manner than healthy cells. So in principle, these are the two components that we would need for our pinads to work specifically in the cells we want to target. And the target we went after is this c-Myc oncogene. This is activated in 70% of human cancers. The protein is too unstructured, so it's too floppy and has too many intrinsically disordered regions so that we can target the protein itself successfully. So a good way to target this protein would be in an indirect manner where we degrade the mRNA of c-Myc, inhibiting the formation of the protein and therefore impacting on cancer and progression. Just quickly, this is our pinad molecule. It binds to c-Myc into these RNA structures that are present in one part of the c-Myc mRNA. These are called G quadruplexes. These are also present in other transcripts as well, in other RNAs, and it binds to copper with very high affinity. So there are the two parts, the RNA binder and the copper recruiter, or the warhead, or the scissors as we call them. And please focus on your attention only on lane number 4. This is a gel where we look at the stability of the c-Myc oligonucleotide. And in lane number 4, we added the c-Myc oligonucleotide, the pinad, the copper, and oxidative conditions. And what we observe is that the c-Myc oligonucleotide is unstable and degrades over time. In lane number 7, we do have the c-Myc oligonucleotide, only the copper recruiter, but not the binder. And what we see under the same conditions, if there is no binding event, is that the c-Myc oligo is completely stable over time. And similarly, if you pre-incubate your c-Myc oligonucleotide with an excess of only the binder, so the binder saturates all the binding sites, then even in the presence of the pinad, because there is no binding event, it has been outcompeted, the c-Myc oligo is fully stable over time. Very important as well is that we require very minimal amounts of copper for these molecules to work. Only with 0.05 equivalents of copper is sufficient to degrade significantly the c-Myc oligonucleotide, which it's a word I don't like using in biology because it's difficult to prove this for example, in a living organism, but there is a potential catalytic nature in the way these molecules could work. So after degrading one c-Myc oligonucleotide, could degrade the next one and in a continuous manner. So we have tested this significantly, and there are certain cancers that are dependent on c-Myc. So if you degrade c-Myc, they will stop proliferating and may start shrinking. And there are other cancers that are known to be non-c-Myc driven. It means that even if you get rid of Myc, they will continue to proliferate. This work is on, just to show it on certain cell lines, we always use as controls the binder alone or the warhead alone, and then we compare with the molecule that has both parts. And what we see is that only the molecule with only the pinad molecule that has the binder and the warhead is able to induce the reduction of c-Myc mRNA. And this is translated, this reduction at the mRNA level is translated into a significant reduction in c-Myc protein in these cells. But while cell models are very useful to a certain extent, animal models can also tell us a bit more because of what Melinda also just explained. There is the tumor microenvironment, there is all these matrix, there are all these different additional levels of complexity. And one thing I always like doing in this project is that I like to start an experiment where we do intratumoral injection to understand the activity of the molecule. And for my surprise, and actually we repeated this twice, so I was really sure about this effect. When we have the binder or the warhead alone, there is no effect on tumor growth. This is the tumor size over days. But when we injected the pinad molecule 4 times with 1-day interval, we got a very strong anti-tumor effect. So this was important. There's an anti-tumor effect, but we need to prove a bit more how these molecules are working. So we took the tumors at day 9 that were very little, and we can look using a Western blot for the amount of protein, of CMYC protein in those tumors. And what we have observed in the pineapple treated group is that the CMYC protein is pretty much absent, which means that we successfully degraded the RNA and stopped the translation into CMYC protein. And this led to a very strong antitumor effect. The same is true when we looked at the tissue by immunohistochemistry, where we see a significant reduction to the CMYC protein. This is not a useful potential treatment or modality to destroy RNA if we cannot dose in a different manner. So we tested as well subcutaneous injection or intraperitoneal injection, IP, and the molecule works very well, again, with the same mechanistic observations. This works and the molecule works well in models that are dependent on CMYC. That's what I'm showing here. This is a gastric cancer model. This is a lymphoma model. And this is to show that there is a dose dependency activity. And importantly, in tumors that are known to be not dependent on CMYC, we don't see an effect. So this is almost looking too good in the sense that tumor cells and tumors that are dependent on CMYC, we have a very strong effect, and we have very little to no effect in tumors that are not dependent on CMYC, showing a very good level of selectivity. We also did a lot of sequencing experiments, and I will be stopping very soon, just to show that we could as well show by 2 different techniques, Illumina sequencing and nanopore sequencing, that we are eating the CMYC transcript, and we have a reduction in CMYC and c-CMYC. And this also, because there are many other transcripts that are known to be dependent on CMYC, we also see a reduction in the ones that should be reduced and an increase in the ones that should be increased. And for our surprise, because G-quadruplexes are present in other transcripts, we also looked into all the different transcripts, all the different RNAs that have G-quadruplexes. And for reasons I don't know to explain completely, because these basothiazoles in principle could also target other G-quadruplexes, we see a very good selectivity for the 5' UTR of CMYC, which now brings these 2 layers of selectivity of the approach. One is the targeting of the mRNA per se, and the other one is the dependency of the modality on copper and on oxidative conditions. Importantly, this also justifies why we do not observe, for example, toxicity for these molecules up to 500 micromolar, for example, in cardiomyocytes or in healthy human tissue. This is not exclusive to CMYC. We can apply to other targets. This is just one different example. So we believe pretty much we have a modular approach here. If we have a very good RNA binder to an RNA of interest that is implicated in disease, we can weaponize that small molecule, that RNA binder, with our warheads in order to destroy that particular mRNA and stop the translation into the protein of interest. I will skip these slides because of time, and the take-home message is really we can create functionality in these silent binders that usually bind, they don't do much, by using these warheads that can cleave RNA in close proximity. We can aim to really develop this approach into making a new therapeutic modality, and I'm very excited because there are many RNA binders out there that have not so much functionality, but they bind to specific RNAs, and hopefully we will be able to explore those using this approach. And finally, this is the most important slide. I want to thank my research group. We are an interdisciplinary research group. We also have a satellite lab in the Oncology Center in Madrid, where we do a lot of preclinical and now also starting some clinical work, and to thank funding from these different sources, and my close collaborators, and in particular my family, for supporting me over these years and sometimes through difficult moments. Thank you so much, everyone.
Thank you very much, and a huge amount of food for thought there. So we're now moving into the question and answer portion of today's webinar. So I'm going to start with a question for Serena. which is back to the thinking about these freeze-dried constructs. So the question is, how does this freeze-drying created collagen structure compares with what's possible with 3D printing, and what's the advantages of either technique?
I think that with 3D printing, then yes, you can produce very controlled and reproducible structures, but you're reliant on building up from a base, I suppose. With the collagen structures, and I'm going to answer somebody else's question at the same time, we can produce fairly large quantities of material in one go. Obviously, the ice nucleation is stochastic in nature, so we probably have less exact control than 3D printing. But actually, we can produce something very, very quickly in a freeze dryer, and our only limitation is the size of the freeze dryer. So we can produce things that are really quite large in diameter, reasonable thickness, and then we can sort of take little samples out of that.
Thank you very much. I'm going to move on to Melinda now. Thinking about how these adapted hyaluronic acids might be administered. So how is that administered? Is it locally? Is it systematically? And sort of linking with that, how close would you get to that effect that you've seen just by injecting normal hyaluronic acid? Would that get you anywhere?
Okay, good question. So these are reactive molecules. They're designed to react as fast as possible when they meet a high concentration of protein, like these fibrous proteins. So they are reactive systemically. They're going to react with proteins in the blood. So they need to be administered locally. And so we're looking at things like arborizing catheters, for instance, to get them into the brain, or obviously tumor resection environments are ideal. You can literally potentially fill the cavity with this drug. It's non-toxic, so it doesn't matter how fast it's released. It's better to be released fast, in fact. Good question about, can you put in normal hyaluronic acid? It needs to be a high molecular weight. Of course, initially it would do the right job, because it will effectively make much higher concentration of hyaluronic acid, and it will do the job of slowing down the motions of the hyaluronic acid and preventing them from flexing enough to get to the cell surface receptors of the cancer cells. The danger is that cancer cells are very, very good at breaking down hyaluronic acid, and so my worry would be that they would very quickly turn that into a hyaluronic acid soup that actually is directing cancer cells to keep going. One of the other advantages of this chemically modified hyaluronic acid is it's quite hard for the cells to break down. And once it's attached to a protein, it's almost impossible. So I won't say it is impossible, because there will be an enzyme out there somewhere, or acid will break it down. It'll stay stuck to that fibrous matrix.
Brilliant, thank you. For Gonçalo, a question around the route to translation for the work that you've got there. Is incredibly interesting science, but how close are we, and how do you get there?
So that's a very good question. And one of the reasons why I do have an additional lab that is more translation is really to be able to push certain technologies to advanced preclinical stages, where we could engage in work with the medical doctors to really bring molecules and approaches, I'll say in the same way as Melinda mentioned, closer to the patients and to the benefit. A typical route that I have done, and we are thinking for this particular approach, is to spin out a small biotech where we can take advantage of the approach, really define one particular particular targets of interest. In particular, I'm very interested in neurodegeneration with this approach, because there are many specific targets on RNA that we could go after, from Huntington's disease to Alzheimer's disease, and that's something we are currently considering. And at the same time, I'm speaking with companies regarding the Hamied program, which I believe it has been showing some good surprises, and sometimes when but maybe there's some problems with this molecule, and then I get another new result that looks positive, which makes me think maybe this approach can help some patients. One of the things we just recently did was to check whether this molecule I just showed you could be, for example, delivered orally, which would be a great benefit in terms of translation.
I am aware of time. I'm going to squeeze one more in. This one's for Melinda. I think probably of interest to everyone. What's the implications of your research for slowing of aging?
That's the $1 million question, isn't it? Well, what we know is that the extracellular matrix changes hugely in aging. And what we know, at least in the vasculature, is that those changes precede the aging of the cells. And that implies that if we can slow the aging of the matrix, we will stop, at least to some extent, prevent some of these aberrant cell behaviors that we see in aging that tend to move towards what we call cell senescence. That's basically the cells still being there, but just doing nothing and chucking out lots of inflammatory factors. What we need to do before we can do anything about that is we need to know how the matrix changes in aging. And bizarrely, we don't. We know lots about it. We know that it gets damaged. We know that it can get oxidized. We know that sugars like glucose react very strongly with it. And those reactions to some extent become irreversible after oxidation. So we know that we know some of the elements, but we haven't been able to put all that together yet. I mean, this is for the next generation of researchers to do. This is why I have that group of people that I pointed out, and likewise you and Serena. I mean, that is a 20-year program to work that out. But now I think it's doable. It is actually doable. And I think it could be done quite quickly if people got behind it.
Thank you. Well, that's a really inspiring note to end on. It's clear today's discussion has sparked a huge amount of engagement, and apologies to everybody for those amazing questions that we haven't had time for today. It's not a conversation with simple conclusions. It's part of an ongoing journey to understand how all these works, and Cambridge is working to transform the future of medicine. So I'd like to just finish by thanking our outstanding speakers, Professor Serena Best, Professor Melinda Duer, Professor Gonçalo Bernardez, for sharing their expertise, giving us a glimpse into the innovations shaping targeted therapeutics and smarter treatments. And thank you to all of you for joining us, for your thoughtful questions, and for being part of this vital conversation about the future of healthcare.